To identify tissue damage in multiple organs that occurred parallel in mice after a mild blast exposure generated in a shock tube, and to assess whether repeated subminimal blasts could have synergistic effects, we used a 3D cryoimaging system developed by BioEnvision Incorporated out of Cleveland, Ohio. This system acquires three-dimensional, very high-resolution, large field of view, bright field anatomy, and fluorescence molecular image volumes from sequential images of the tissue block face by alternately sectioning and imaging the whole mouse. With this system, we have anatomically characterized in 3D, from whole animal to tissue level, the subtle changes caused by blasts. Figure 1 shows representative orthogonal slices of the mouse brain 24 hours post-exposure to a single mild blast, shown here as the A group of images, as well as to repetitive subminimal blasts, shown in B. White arrows in B point to petechial bleeds, or pinpoint hemorrhages, observed throughout the brain tissue after repetitive subminimal blasts. In contrast, no such petechiae were observed after single mild blast. Figure 2 here shows representative orthogonal slices of the lungs post-exposure to single mild blast and repetitive subminimal blasts. Clear differences can be seen in the lungs, with the dark red lung color of the repetitive injury mouse in B, indicating extravascular blood that was unable to be cleared during the perfusion process, while the lungs of the single mild blast mouse in A also appear to demonstrate petechial hemorrhages. The number of petechiae appears to be higher after repetitive subminimal injury. The 3D cryoimaging system uses a specialized multi-scale visualization scheme, which provides color-rich volume rendering and multiplanar reformatting, enabling one to visualize an entire mouse and zoom in to organ, tissue, and microscopic scales. These video clips show orthogonal and sagittal slices taken in consecutive order in mice subjected to repetitive subminimal blasts 24 hours post-exposure. Changes seen in multiple organs parallel clearly demonstrate the polytrauma nature of mild blast-induced changes.